Hi, this is a lecture for my anatomy and physiology students, and this is the second part of natural resistance. All right, we are continuing talking about the components that play a role in natural resistance, and now we're going to talk about physical and chemical barriers. And I lumped those two together because it kind of plays a role in both skin and mucous membranes. So we're going to talk about how skin plays a role first. So first of all, how does skin play a role in our natural resistance? It is a barrier. It is one of those barriers, and it plays a role in both being a physical barrier and also components of it will play a role in chemical barriers. So physical barrier first. So the skin, it is very dry. It's a dry environment, and most pathogens don't like that type of environment. It's too dry and poor in nutrition for a lot of growth. We do have growth of bacteria on the surface of our skin. It contributes to our microbiome, but that is for another day. All right, so our skin is also made up of closely packed keratinized cells. We've already talked about this as far as the integumentary system goes. We know that there are cells of the skin that are tied together with tight junctions. And so there's not a whole lot that gets between those cells. So it creates that physical barrier. The cells are keratinized, so it makes them able to withstand abrasion. It also builds upon that barrier. Now, skin has sebaceous glands, and sebaceous glands secrete sebum. Sebum makes its way to the surface of our skin and coats our skin and also our hair. That sebum contributes to that barrier. It is part of our barrier. It creates this protective film, and sebum in its makeup is also a little bit of bacteriostatic, so it helps to keep the growth down. All right, and then we also have sweat glands that release sweat, and sweat has a pH that's on the lower side, so anywhere from 3 to 5 on the pH scale. And there's also salt in the pH. So a lower pH, or no, a lower pH and salt in the sweat, and that creates this environment that not a whole lot of pathogens like, and not they don't like to. There's not going to be a whole lot of growth there at the skin. All right, so on to mucous membranes, and we're going to talk about just a few of the mucous membranes and how they contribute. We're going to first start off with the lacrimal gland in the eye. So the lacrimal gland produces lacrimal fluid. We know this as tears. Now, in that fluid, what does it do? It's used to wash away debris and maybe even pathogens. Now, lacrimal fluid does contain lysozyme. Lysozyme, Lysozyme, and that is a compound that is bacteriostatic. So it helps to prevent the growth of bacteria, just mild bacteriostatic. Um, so on to our salivary glands, thinking about our oropharynx. In our oral mucosa, we find the salivary glands dump into our oral mucosa, and it dumps the saliva. So our saliva also has lysozyme in it, which is mildly bacteriostatic. All right, so onto the mucous membranes of the respiratory passages. So our respiratory system starts with our oropharynx, our nose, onto our pharynx, all the way down our trachea to all of the bronchi and all of the other respiratory passages. So all of that is lined in a mucous membrane which secretes mucus. That mucus is very sticky. So when we breathe in air, yes, we want air to come in, and the ultimate goal is to have air exchange in the tiny air sacs in our lungs. However, with our air comes lots of different particles, particles that we typically can't see because they're microscopic. So dust, bacteria, other droplets, um, parts of insects, all of that we're breathing in, and that debris will get stuck in the mucus. Now, what is typical is the macrophages are there and they will usually gobble up and destroy any type of bacteria or debris that happens to get in, and that helps us with our natural resistance. However, the cells that line our respiratory passages are also ciliated, so they have cilia. And the picture right in the middle is a picture that shows the mucus with underlying cilia. So those cilia all beat in the same direction. So they're all beating up and out in the same direction. So as you can imagine, when, let's say, a piece of dust that could be carrying bacteria, a lot of times it is, lands and gets stuck in that mucus, yes, the macrophages can come and eat it and destroy it. However, because of that cilia beating all in the same direction, it's going to move it up and out of those, physically move it up and out of the respiratory passages to a point where you can get it to your throat. We call this the mucociliary escalator. So escalator, moving it up and out. So we can, sometimes you have kind of a tickle in your throat and you go, clear your throat. And what you're doing is you're clearing that stuff that was brought up through your respiratory passages, and either you can spit it out or you can swallow it, and you're getting it up out of that respiratory passage, which is the goal, right? Because we want only air exchange, we want it clear for air exchange. So our mucociliary escalator helps us trap and move that debris and potential pathogens up and out of our respiratory passage. Okay, one more example given the mucous membrane. Our digestive tract is also lined in a mucous membrane. So it secretes that mucus. Also think of it being this slime layer that helps move the food through the digestive tract. Now, let's just think about the stomach for a minute. There are cells that line our stomach, that make up our stomach wall, that secrete hydrochloric acid. Yeah, hydrochloric acid, and it contributes to the low pH of our gastric juice. We also secrete various enzymes that are secreted as part of our gastric juice. Our gastric juice has this low pH and these enzymes that will break down certain compounds. So if bacteria and other pathogens get into our stomach, a lot of times that gastric juice will break them down and render them not no, that doesn't sound right. It will break them down, therefore they aren't a threat to us anymore. Now there are some bacteria and viruses and other types of pathogens that can withstand our gastric juice and do, and eventually cause us problems, but the gastric juice will help us with our natural resistance, keeping those numbers down. Okay, so it attempts to, it tries to, doesn't always, it's not 100% fail safe. Now we're on to cellular defenses, and these are the cells that help us with our natural resistance. Overall, these are the white blood cells, so leukocytes. We can break down leukocytes into two major groups, granulocytes and agranulocytes. Granulocytes contain granules, okay? And on your flowchart there on the screen, they've listed 3 granulocytes. Those are the neutrophils, eosinophils, and basophils. Neutrophils specialize in destroying bacteria. Eosinophils specialize in destroying worms and worm eggs. Basophils specialize in releasing those granules and with increasing the histamine response. So produces inflammatory and allergic reactions. So it's going to increase our inflammatory response by releasing histamine. Now, our granulocytes are the lymphocytes and the monocytes. Lymphocytes, there's a few different types of lymphocytes. We have B cells, T cells, and natural killer cells. As far as the natural resistance goes, natural killer cells are the only lymphocyte that we're really going to talk about for natural resistance. All right, so the natural killer cells. So we know that natural killer cells are a type of lymphocyte. And you may not have heard about natural killer cells before. They are pretty cool. And these are cells that can move around in our body. So they can move in and out of the bloodstream, they can move around in our body, and they're constantly on patrol. So they're always on surveillance. They're good for surveillance. And what they will go up to a cell and they will scan, think of it, it's like they scan the surface of that cell. And whenever they encounter a cell that has abnormal or unusual plasma proteins, they kill it. So they can recognize a cell that's not supposed to be in our body. It's pretty wild. And they recognize it by scanning the surface, looking at the proteins on the surface of that cell. And if they don't recognize the set of proteins, then they destroy that cell. They're also called lethal lymphocytes. Now you know why. And they kill indiscriminately. So what I mean by that is they don't stay around to ask, well, wait a minute, I don't recognize you. Who are you? And what do you want? They don't do that. They recognize it as being not, they don't recognize it. They don't recognize that pattern. They kill it. So you think, okay, well, how do they kill another cell? How does one cell kill another cell? They use what are called granzymes. And these are molecules that will poke holes in membranes. Okay? So think of these granzymes kind of like knives. All right, so they're going to take these granzymes and poke holes in in the cells. So take a look at the picture that I have right here on the bottom of the slide. And so natural killer cells will typically kill viral infected cells and also cancer cells. Both virus-infected cells and cancer cells will have plasma proteins that have changed, that are different. So the natural killer cell will not recognize it as being a normal body cell. So it's kind of cool for us, right, to have these natural killer cells. We definitely want them as part of our arsenal because they do kill cancer cells and they do kill virus-infected cells. Another way to think about how they do this is with grenades. So you can think of these granzymes as being grenades. That might be an easier way for you to remember it. So you have, if a natural killer cell doesn't recognize it, right, it's going to take a grenade, a granzyme, and toss it into the cell, and it's going to cause that cell to die. And we call that apoptosis, programmed cell death. So that's probably a pretty good way to visualize that. Granzymes being like grenades, throwing it into the tumor cell, throwing it into the virally infected cell, and causing those cells to die. All right, now we're going to talk about phagocytes. So what is a phagocyte? A phagocyte is a type of white blood cell that uses phagocytosis to engulf bacteria, foreign particles, dying cells, all to protect the body. Okay? So right now we're thinking of pathogens. So phagocytes that engulf and destroy pathogens. There are white blood cells that are considered professional phagocytes because they do such a good job at phagocytosis and they're very efficient at it. There's 5 of them. There are neutrophils, monocytes, macrophages, dendritic cells, and mast cells. These are all types of white blood cells that are known as professional phagocytes. So now I want to go over the process of phagocytosis. So that's the next thing we're going to do, go over the process of phagocytosis. All right, so the process of phagocytosis. And I have it listed as 1 through 4 because you know I like to list things. All right, so as we're going to be talking about this, let's imagine a macrophage. And the picture is showing a macrophage. Let's imagine a macrophage and it is, and its target is a bacterial cell, okay, the pathogen, a bacterial cell. Now, how does the macrophage know where the bacteria is? How does it know where to go? Because macrophages can move around in and out of tissues throughout our body. How do macrophages know where to go? What they do is they will move towards that pathogen. They will follow a chemical trail, and we call that chemotaxis. So the process by which a macrophage finds the bacteria or another pathogen is by chemotaxis. It follows a chemical trail. Now, once it finds it, it's going to adhere to the bacterial cell. So the macrophage adheres to the bacterial cell. How is it really adhering? It's recognizing and binding to a molecular pattern on the surface of the pathogen. Okay, then it has to ingest. It has to move that cell from outside the macrophage to inside the macrophage. We call that ingest. And the process by which that happens is endocytosis. You guys have already learned about the process of endocytosis. And so think about that process of endocytosis, take a look at the picture that's up on the screen, probably remembering that process, helping to remember that process. That bacteria is going to be moved in and it's going to be wrapped in that vesicle. However, we call it now the phagosome. Okay, so we're calling it the phagosome. All right, number 4. Now what happens is that macrophage has to destroy the bacteria. It can't just keep it inside. It's going to have to destroy it. And how it does that is with the help of some molecules that are inside of a lysosome. So the lysosome is going to merge with the phagosome, and as a result, it's going to release free radicals and enzymes and cause an oxidative burst at the bacteria. So free radicals are types of toxic oxygen and also containing enzymes to help break it down. The result of that is the bacteria gets broken down into tiny pieces. We're breaking it down and therefore killing that cell. So that's how we say it digests it. It breaks it down into smaller pieces, breaks that bacteria down into smaller pieces and therefore kills it. So of phagocytosis, there's 4 major steps: bind, adhere, ingest, and digest. All right, so now we just learned how macrophages do their jobs, right? Macrophages are pretty cool. They're one of my favorite types of cells. There are 2 major types of macrophages. There are wandering macrophages and fixed macrophages. Wandering macrophages, what they do, they wander, right? They have the ability to travel throughout the body, throughout different tissues and throughout the body. Fixed macrophages tend to stay in one type of tissue. And because they do that, we give them special names depending on where we find that macrophage, or basically in which type of tissue do we find that macrophage. And they could be specialized for that tissue. And that's typically what we see. All right, so I'm going to give you some examples of types of fixed macrophages. The type of fixed macrophage that we find in the liver is called a histiocyte. Okay, there is one found in the lung. It's called alveolar because that's typically where we find it, in the alveoli. So an alveolar cell is a macrophage that lives in the lungs. It used to be called a dust cell, but now we call them alveolar cells. Okay, another one is a macrophage that's found in the liver, and we call that a stellate. An older term for that is a Kupffer cell. Okay, the fixed macrophage that is found in bone, I know you'll recognize this one, is an osteoclast. So an osteoclast is the fixed macrophage that's found in bone. And in the fixed macrophage that we find in the central nervous system is the microglial cell. Now, remember I said the fixed macrophages can become specialized for that particular type of tissue. They stay there, they're within that tissue, and they do become specialized to that tissue. So macrophages do help us. They help us with our natural resistance, and they are pretty cool and my favorite type. All right, so that is it for the second part of the natural resistance, and thank you for paying attention.